Protein synthesis is the process of forming proteins within cells using the code found in DNA. The code is determined by the sequence of bases on the DNA strand. It is made up of two steps, transcription and translation. In the process of transcription, code from a DNA strand is copied to a strand of mRNA, which is a messenger RNA that carries the base code to a ribosome to synthesize a protein. The process of transcription occurs within the nucleus, as that's where the DNA is, and that's where the mRNA begins to copy the base code. The enzyme known as helicase within the nucleus unzips the DNA strand, giving the mRNA a chance to copy the base code. It separates the bases already on the DNA strand by breaking their hydrogen bonds. Nuclear pores on the nuclear membrane allow for nucleotides from the cytoplasm to enter into the nucleus. Once in the nucleus, DNA polymerase attaches these nucleotides, and on these nucleotides form these bases. They're complementary bases to one of the DNA strands that the mRNA is bonding to. And it's important to note that the T base does not exist on the mRNA strand, but is replaced with the U base. The U base is represented by our pink bases. Once the mRNA strand has finished copying the code of one of the DNA strands by forming its complementary bases, the enzyme ligase solidifies the phosphate sugar backbone on the mRNA, and the mRNA leaves through nuclear pores out into the cytoplasm, where it prepares to go under translation. With the mRNA now complete, the DNA previously unzipped by helicase zips itself back up and forms its double helix once again, with its complementary bases being bonded through hydrogen bonds. In the process of translation, the mRNA previously formed during transcription goes to the ribosome made up of rRNA in order to synthesize proteins. Currently in the cytoplasm, mRNA heads to the ribosome, made up of ribosomal RNA, also known as rRNA. Also in the cytoplasm is tRNA. tRNA attaches itself to an amino acid present in the cytoplasm, and each tRNA has a specific set of 3 bases called an anticodon. As the mRNA enters the ribosome, sets of 3 bases on the mRNA called codons code for a specific tRNA with this matching anticodon with the complementary bases. The first codon on any mRNA strand that is read will be AUG, as this calls for the methionine amino acid to be pulled into the ribosome and begin the amino acid chain. Once the methionine amino acid has bonded, the following codons are read, and the corresponding tRNA with the correct anticodons will be pulled into the ribosome, and methionine will transfer to these amino acids. This sequence continues until the amino acid chain is complete. As new tRNA is pulled into the ribosome and bonds to the mRNA, the amino acids transfer to that tRNA, leaving the empty tRNA to exit into the cytoplasm to gain another amino acid. This sequence continues until the amino acid chain is complete and a codon for stop has been read. In this case, we have UGA, and the amino acid chain will then pop off as there isn't another tRNA that is pulled into the ribosome. At this point, the amino acid chain is complete, and so the mRNA is no longer being used, and so it is recycled back into the cell for its parts to be reused. At this point, the amino acid chain has completed, and the amino acids are bonded through peptide bonds. This forms a polypeptide, otherwise known as a protein. The amino acids that make up this protein and the shape of the protein determine its function either in the cell or somewhere else in the body. In this case, this protein was formed in a free ribosome, meaning the protein will be used within the cell. However, if it was formed in an attached ribosome on the rough ER, it would be sent to the Golgi to be customized and shipped somewhere else in the body. That is the complete process of protein synthesis, from the original DNA code coding for a specific protein, all the way to that protein synthesis.